Hi, I'm Sam Horsfield from Imperial College London, and I've been using PopPUNK to compare evolutionary characteristics of diverse bacterial pathogens. So PopPUNK is a tool that enables estimation of core and accessory divergence in bacterial species in huge genomic data sets. It does this by using variable length k-mers and calculating matches of these variable length k-mers between pairwise comparisons of genomes. If you look at the relationship between the accessory and core genome divergence across different species, you can see that it's highly characteristic of different bacterial populations and species. And this is due to underlying characteristics in population structure, which is a product of evolutionary trajectory and epidemiological factors. So the aim of this work is to use the core and accessory genome relationship calculated by PopPUNK to see if we can identify a signal of selection in the form of the accessory over core genome divergence, or A over pi, assuming neutrality. And the idea of this is to see whether we can detect selection in the accessory genome and answer the question of how the accessory bacterial pangenome, or the accessory genome in bacterial pathogens evolves. So to do this, I developed two methods. One is an analytical method, which uses a derivation of the Dukes-Cantor model of base substitution. And the other one is a simulation-based method using approximate Bayesian computation using the tool ELFI. The reason we need to use these approaches is because PopPUNK estimates distances in the core and accessory genome. And this is actually an underestimation of the true rate of divergence in those two genome compartments, because ancestral mutations along lineages can be overwritten by multiple events at the same site. And so this can cause underestimation of your accessory or of your distances. So I have data sets from 20 bacterial species, ranging in size from 1,000 genomes up to 50,000 genomes across 6 phyla. And this is just a snapshot of 4 data sets and the core and accessory relationship between them. So in red is TB, and in blue is Pseudomonas aeruginosa. You can see there's a massive difference in the scale of diversity in these 2 bacterial species. And also comparing, say, Streptococcus pneumoniae and Staphylococcus aureus. These are more similar in sort of the scale, but also have very different distributions in terms of their core and accessory relationships. So the idea here is to sort of come up with models that are flexible enough to be able to calculate a relationship between the accessory and core divergence. Here I've generated simulations that underlie both the analytical and simulation-based method, and trying to see whether I can match expected relationships between the real rate of divergence that's underlying these models, and also then the distances that are calculated by PopPUNK. And as you can see here, there's an exact match in both the core genome and the accessory genome. But also, the simulation is very flexible, and you can play around with the parameters, and you can fit all kinds of different relationships between the core and accessory genome by varying these parameters, such as gene gain and loss here, and per base substitution rate up on the y-axis there. So these 2 panels show the results from the 2 different models. So for the analytical fit, these are PopPUNK plots. So the core distances on the x-axis and the accessory distances on the y, and then the analytical model fit is the black line connecting them. And as you can see, there's quite a good fit to all of these data sets. And you can also, as one of the parameters that's optimized, get an estimation of the rate of accessory divergence over the core, assuming neutrality. And that's sort of what we're after here, to see how the accessory genome is evolving. And these are the results here for the analytical approximate Bayesian computation model, the simulation-based model. And these are the marginal posterior distributions for the accessory over core divergence, or A over pi. Again, they're estimated. The thing to note about this is that these 2 models are estimating different values for the accessory over core divergence. There's 2 potential reasons for this. Potentially that the models aren't correct, which is potentially likely. And this is due maybe because the analytical model is too flexible, or the ABC model isn't flexible enough. The other potential reasoning for this is that a model of neutral selection isn't actually correct when you're trying to determine the effect of, or how the accessory genome diverges relative to the core. So that could be the other reason behind that. So future work will look to sort of further iterate these models, potentially decreasing the flexibility of the analytical model by reducing the number of parameters that are being estimated, and potentially increasing the flexibility of the ABC model by increasing the number of parameters being estimated. Further work, again, on top of this, will model a non-constant relationship between the accessory and the core genome divergence, and this will factor in whether or not selection is occurring in the accessory genome. Thank you very much.